after replication, the next process is transcription, and transcription process also called heterocatalytic, because here from DNA to RNA synthesis takes place, mRNA, right? Because we know from DNA the next part is mRNA will synthesis, right? From DNA to DNA synthesis we know replication, we have learned this part. Now DNA to mRNA synthesis is called the transcription, and it is also called heterocatalytic. DNA will help in the synthesis of mRNA, and this will help in protein synthesis, because from mRNA protein synthesis will now takes place, that we know. So for these things, first we have to learn something fast. Suppose this is the double strand DNA, 3' to 5' end, and obviously this is antiparallel, so this will be 5' to 3' end. Suppose here are some codes I have written here, opposite codes I have written here, T A A T G C C G G C A T T A A T, right? So this is the, and we know the 3 codons responsible for synthesis of a particular amino acid, or 3 or 1 codon is codes for a particular amino acid. Now we know from DNA mRNA will synthesis, right? If here the one strand of mRNA is synthesis, so what will be the code? A will code for U, because in RNA T is not there, so A will U, T A C G G C C G T A again U T A U. Why? Because this is RNA, so for RNA uracil, not thymine, right? This is codes. But you see one thing, we call this strand is coding strand, and this strand is the template strand. Template strand, the strand which should be transcribed in the next part. If we take, if A codes for U, T codes for E, this is the coding strand. Okay, so now you see the opposite thing, T is not code for U, A is not code for A, so these are not making the similar thing, right? So what we will take, we will take this we will take as a codon strand. Okay, suppose this is mRNA synthesis, this will act as a template strand, so opposite base pair we will take T A A U. For this opposite thing we will take T A A U G C C G G C A U T A, because this is RNA, U will be there, T A A U. So you always remember 3' to 5' end is always act as a template strand, and 5' to 3' end always act as a codon strand. You have to coding strand, you have to remember this. Okay, so the enzyme which help in RNA synthesis is called the RNA polymerase, and in prokaryotic only one RNA polymerase is there, but only one RNA polymerase. But in case of eukaryotes, one RNA polymerase, and we know the RNA, tRNA, mRNA. But in eukaryotes there are RNA polymerase are 3 types: RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. And here this only one RNA polymerase is responsible for synthesis of RNA, tRNA and mRNA. But here you see RNA polymerase 1 responsible for synthesis of rRNA, RNA polymerase 2 responsible for synthesis of mRNA and hnRNA. We know from hnRNA mRNA form, and RNA polymerase 3 is responsible for synthesis of tRNA. Clear? So what I have tell you, ki from 3' end to 5' end always you remember this act as a template strand, the strand which should be transcribed in the next generation, and these strands opposite codon will be এই স্ট্যান্ডটা থেকে অপোজিট কোডসগুলো নিয়ে এমআরএনএটা সিনথেসিস হবে, এই স্ট্যান্ডার্ড কোডগুলোর অপোজিট কোড নিয়ে হবে না। তাই জন্য আমি এইটা যেটা ড্র করেছি, দিস উইল নট টেক প্লেস, দিস উইল টেক্স প্লেস। ক্লিয়ার? সো ফ্রম 3' to 5' end the mRNA will synthesis, sorry, yeah. So 3' end to 5' end, this is called a template stand, and 5' end to 3' end will call the coding stand. Okay, so RNA polymerase is made up of, the structure of RNA polymerase we will learn now. It is made up of 5 subunits. The first 2 alpha subunits helps bind to the initiation protein, binds to initiation protein, the 2 alpha subunit bind to initiation protein, 1 beta dash subunits bind to DNA template stand part, 1 beta subunit, it is beta dash and which is beta, bind to nucleotides, and other one is 1 omega subunit. And these 4 subunits, 1 2 3 4 5, these 5 subunits are called code enzymes. Okay, and there is another associative 2 subunits are there, that is called sigma. This is the sign of sigma, and rho factor. Sigma is responsible for initiation of RNA chain, and rho factor is responsible for elongation and termination. We will see their function when we will see from DNA how mRNA is synthesizing. These are temporarily attached, and these are the permanent subunits. Okay, acha. So first we have learned, this is the DNA, double strand DNA. This is the 3' end to 5' end. Obviously the opposite will be 5' to 3' end. And in the 3' to 5' end, we always know at the 3' end OH group is there. So RNA polymerase, whatever the thing will take place at the functional site, so RNA polymerase will bind at the 3' end. Suppose this is the RNA polymerase. Okay, acha. So now here there is a promoter part. Promoter, it's normally a sequence of nucleotides. Promoters is normally a sequence of nucleotides. Here TAA, in this way the nucleotide will go, and this is called the Tata box. So here will be the promoter, and promoter is very responsible for a primer. Suppose promoter will help to formation of a primer. Acha, what we will সাপোজ আমাদের বাড়িতে যখন রং হয় তার আগে কি করা হয়, একটা প্রাইমার দেওয়া হয়। প্রাইমার কি করে, কোনো কিছুকে বাইন্ডিংটাকে মজবুত করতে সাহায্য করে। সেরকম প্রোমোটারও কিছু প্রাইমার সিনথেসিস করবে, সেই প্রাইমারগুলো এই স্ট্রাকচারটাকে বাইন্ড করতে ভালো করে সাহায্য করবে। আচ্ছা, প্রোমোটার মানে কোনো কিছু, এই যে mRNA সিনথেসিস হবে তার জন্য একটা প্রোমোটার রিজিয়ন চাই যেখান থেকে জিনিসটা স্টার্ট হবে। সো প্রোমোটার উইল বাইন্ড অ্যাট দ্য 3' end and also with the 5'. So promoter, you see, attach 5' end of coding stand. This is the coding stand, this is the template stand. As I told you, 3' to 5' end, 3' to 5' end is the template stand, 5' to 3' is the coding stand. So here this is the template stand and this is the coding stand. Clear? So this promoter attach 5' end of the coding stand and 3' end of the template stand, 5' end of the coding stand, 3' end of the template stand, and whatever the thing, anything which is at, just when something is towards 5' end is called the upstream. যখনই কোনো কিছু 5' এন্ডে এসে বাইন্ড হয় সেটাকে আমরা বলি আপস্ট্রিম, আর যখনই কোনো কিছু 3' এন্ডে এসে বাইন্ড হয় সেটাকে আমরা বলি ডাউনস্ট্রিম। সো সেই জন্য আমরা এই যে 5' এন্ডে যে প্রোমোটারটা বাইন্ড হলো সেইটাকে আমরা বলবো আপস্ট্রিম, আর 3' এন্ডে যদি কোনো প্রোমোটারটা বাইন্ড হয়েছে তাই এইটাকে আমরা বলবো ডাউনস্ট্রিম। ক্লিয়ার? দ্য নেক্সট স্টেপ, হোয়াট উইল হ্যাপেন? In the next step, the terminator will bind. Terminator is towards the 3' of coding string. You see here is a terminator. It will terminate, means when mRNA synthesis will not continue. Okay, we will see. So there will be a terminator. It is at the 5' end of the template stand and 3' end of the coding stand. Or anything which is bind at the 5' end we call this the upstream, and anything which binds at the 3' end is called the downstream. So here I have written the terminator is towards the 3' of the coding stand, and coding stand 3' end means this is downstream, and 5' end of the template stand, this is upstream. The terminator binds in this way. So we know coding stand always 5' to 3' end, and template stand always 3' to 5' end. We have already seen here 3' to 5' end is the template stand, 5 prime to 3 prime end in the coding stand. Now RNA polymerase binds at the RNA polymerase binds at the 3 prime end of the template stand. Okay, when RNA polymerase this binds, some nucleotides will start synthesis. We know in RNA polymerase there is some factor. The sigma factor will bind here, and as we know, it is responsible for initiation of RNA chains, right? So when RNA formation will takes place, the sigma factor is responsible, because we have learned its function, responsible for initiation. এটা শুরু করতে এই RNA মানে DNA থেকে তো RNA ফর্ম হবে, mRNA চেনটা সেটা শুরু হতে এই সিগমা ফ্যাক্টরটা লাগে। তো অবভিয়াসলি সিগমা ফ্যাক্টরটাকে এখানে বাইন্ড হতে হবে, তো সিগমা ফ্যাক্টর বাইন্ড হয়েছে, তো এখানে কিছু এবার পরপর কিছু নিউক্লিওটাইড ফর্ম হবে। যখনই এই RNA পলিমারেজটা এখানে স্টেবল, এখানেই ফিক্স থাকবে না, এটা এদিকে মুভ করতে থাকবে। তো এই RNA পলিমারেজ যখন এখান থেকে এখানে মুভ করবে, মানে 3 প্রাইম এন্ড থেকে 5 প্রাইম এন্ডের দিকে মুভ করবে, তখন এই সিগমা ফ্যাক্টরটা পড়ে যাবে, মানে ফল অফ। এটার আর কোনো কাজ নেই, কারণ এ তো ইনিশিয়েশন শুরু করে দিয়েছে, ওর কাজ শেষ। এবার আমাদের কোন ফ্যাক্টর আসবে? রো ফ্যাক্টর। মানে এরকম পরপর কিছু হতে থাকবে। রো রেসপন্সিবল ফর ইলংগেশন অ্যান্ড টার্মিনেশন। ইলংগেশন মানে mRNAটাকে ইলংগেট করতে এই রো ফ্যাক্টরটা দরকার। সো এবার RNA পলিমারেজটা এখান থেকে যখন এখানে এলো, তখন রো ফ্যাক্টরটা এর সাথে অ্যাটাচ হলো। রো ফ্যাক্টরটা কী করলো? mRNAটাকে নতুন আরও নিউক্লিওটাইড এসে যোগ করলো, mRNAটা ইলংগেট হলো। আর এবার কী হবে? ওটাকে টার্মিনেটও করবে। টার্মিনেটারের জন্য আমাদের কী রয়েছে? টার্মিনেটর রয়েছে, যেটা আমাদের 5 প্রাইম end of the template stand at 3 prime end of the coding stand already present রয়েছে। তো সেটা এখানে ওটাকে টার্মিনেট মানে mRNAটাকে স্টপ করে দেবে, তুমি আর সিনথেসিস হলো না। অ্যাকচুয়ালি দিস ইজ নট mRNA, দিস ইজ hnRNA. We already learned from hnRNA how mRNA synthesis takes place. We already learned these things here, right? So this is hnRNA. I told you there is some intron and exon. Exons are the coding part, introns are the non-coding parts. We know that we want the coding part, so the intron exon will separate by an enzyme called endonuclease. The process is called splicing. Then the exons will now join, right? And this is not the proper structure of mRNA. In the cap and the cap and the tail will join here, right? At 5 prime to 3 prime in the 5 prime cap and the 3 prime in the tail will join, right? And the structure of mRNA is this, right? So here 5 prime in the cap, 3 prime in the polyadenine group, and this is start codon, this is stop codon. And always I told you from start codon there will be a code AUG, and this is for methionine amino acid, right? So we already learned that thing. Acha. So in this way hnRNA will synthesis, from hnRNA mRNA will synthesis. So this is the process called transcription. So this mRNA will help in the synthesis of protein. We will see in the part of translation. So what is the process? Steps. First, promoter recognition. We have learned that promoter is bind in the 3 prime end of the template stand and 5 prime end of the coding stand. And which enzyme helps here? RNA polymerase with the sigma factor. Then elongation, new nucleotides come here and the chain will elongate it. Which enzyme will help here? RNA polymerase, but it's rho factor, right? Rho factor is responsible for termination and elongation. Acha, then termination, again the rho factor helps in termination, right? And again here is a terminator portion, so terminator will stop the synthesis of mRNA. We don't need a much longer mRNA, a particular mRNA we need. So the terminator or RNA, in RNA polymerase the rho factor is there. This will also terminate the formation of mRNA. So this is the process of transcription.